In this video, I'm going to talk about lipids, which are one of the four important biological molecules. So in terms of their structure, lipids, like the other organic biological molecules, have a carbon skeleton base. And this carbon skeleton base has many non-polar covalent bonds in it. Lots of carbon-hydrogen bonds and lots of carbon-carbon bonds. And these are nonpolar bonds, which means they don't carry any partial charges. Lipids characteristically have very few polar bonds as a part of them, or few charges, but there are a couple exceptions to this rule. So because the lipids have so many nonpolar bonds, overall as a group they're nonpolar, and of course substances that are nonpolar are generally hydrophobic. Again, there's an exception to this rule, and we'll look at that in a little bit. When it comes to that monomer-polymer pattern that we see with other organic biological molecules, such as carbohydrates, proteins, and nucleic acids, there is no true monomer or polymer here with lipids. Some textbooks will say that fatty acids are a monomer to lipids, but that's only applicable in certain cases. So we're just going to say there is no monomer-polymer pattern here. Examples of lipids include triglycerides, which are also known as fats. Triglycerides are really important for storing energy in the human body. Another example of a lipid is a phospholipid. Phospholipids are really important for forming the outer layer to cells called the cell membrane. Another example of a lipid is something called a steroid. So a steroid is a group of lipids that includes things like cholesterol, testosterone, and estrogen. And these lipids are really important for making certain types of hormones, but they have other roles in the body too. In terms of nutrient sources for lipids, obviously foods that are high in fat are going to be high in lipids because lipids are a type of fat. So certain types of meats are going to be high in lipids, but there are certain vegetables like avocados, for example, that also contain important lipids. Let's take a closer look at lipid structure. So as I mentioned before, all lipids contain multiple carbon-carbon, carbon-hydrogen bonds. These are nonpolar bonds, which makes lipids hydrophobic. Many lipids, but not all of them, also contain a substructure called a fatty acid. A fatty acid is also known as a hydrocarbon chain. It's a long chain of carbons with hydrogens attached to it, as the name hydrocarbon would suggest. Fatty acids can come in two types, if you will, or two categories: saturated or unsaturated. A saturated fatty acid contains no carbon-carbon double bonds, whereas an unsaturated fatty acid will contain carbon-carbon double bonds. And these carbon-carbon double bonds are important because they cause bends in the structure of the fatty acid, so it makes a difference in terms of the structure of the fatty acid, and of course structure is function, so these unsaturated fats have a different function in the body than the saturated fats. In these pictures here, we see three examples of lipids. At the top of the screen, we see a triglyceride. A triglyceride contains three fatty acids. In the middle of the screen, we see a phospholipid. A phospholipid, like a triglyceride, also contains fatty acids, but it only has two fatty acids, not three. In place of the third fatty acid, the phospholipid has a phosphate group, and that's where it gets its name. And at the bottom of the screen, we see a steroid. And steroids obviously look very different than the other two types of lipids. And that's because steroids contain ring structures. So rather than having a long chain of carbons, the steroids have rings of carbon that are reminiscent of a carbohydrate. But notice the lack of oxygen in the steroid. There's almost no oxygen here. So let's take a closer look at the triglycerides. So tri means three, right? And what do triglycerides have three of? They have three fatty acid chains. These three fatty acid chains are attached to a carbon skeleton that's called glycerol. So the glycerol acts as the backbone to the triglyceride, and then the fatty acids attach to that glycerol molecule. These are really excellent for energy storage, and that's because there's actually energy found in those carbon-carbon and carbon-hydrogen bonds in the triglyceride. So we're going to store triglycerides in the body for energy. We store these in tissues called adipose tissues, and the type of cell that stores these triglycerides are called adipocytes. Triglycerides are very, very, very nonpolar. Right? There's almost no polarity anywhere on them, and therefore they are extremely hydrophobic. Next up are the phospholipids. Phospholipids, as I said before, are very similar to a triglyceride. Like a triglyceride, they have a glycerol carbon skeleton backbone to them. And like a triglyceride, they contain fatty acids, but instead of 3 fatty acids, the phospholipid only has 2 fatty acids. And in place of the third fatty acid is a group containing phosphate. So in the picture here, we can see our 2 fatty acid chains that are part of the phospholipid. And then over on the left-hand side of the screen, we can see that phosphate group. On the very most right-hand side of the screen, we can see what's known as an icon model of a phospholipid. It's just that little circle with the little tails coming off. And that's a pretty common representation for a phospholipid that we will see throughout this class. The phosphate portion of the phospholipid is referred to as the head of the phospholipid, whereas the fatty acids are called the tails of the phospholipid. So the 2 fatty acids, those tails, they're very hydrophobic because they contain mostly just nonpolar carbon-carbon-carbon-hydrogen bonds. But the phosphate group is extremely hydrophilic. It's both polar and charged. So here we have a single molecule, a phospholipid, that is both hydrophobic and hydrophilic. And this is not very common. This property of being both hydrophobic and hydrophilic is called amphipathic. So the amphipathic nature of phospholipids is really important for the function of phospholipids in the human body because it allows the phospholipids to form structures called phospholipid bilayers, which will form the basis to that outer layer of a cell called the cell membrane. If you put pure phospholipids into water, they will automatically aggregate into certain shapes. They'll either form a phospholipid bilayer where the hydrophobic tails are facing one another and the hydrophilic heads are facing the watery environment. Or they might form a structure such as a micelle, which is sort of like a ball with all of the phospholipid heads facing the outer watery environment and all the tails facing the center of the sphere. The last lipid we're going to look at are steroids. And as I mentioned before, the steroids look very different compared to the other lipids like triglycerides and phospholipids. They don't have any fatty acid chains to them. And they do have multiple carbon-carbon-carbon-hydrogen bonds, making them very hydrophobic. But these carbon-carbon bonds are found in ring structures rather than in long chains. So a steroid typically has 4 ring structures and then some other groups attached to it. Examples of steroids include cholesterol, which is really important in the cell membrane of cells. It also includes many hormones like estrogen, testosterone, and cortisol. Not all hormones are made from steroids, but many of them are.